Hi, this is Miss Litton, and this is my wonderful period 5. Say hi.
Hi.
And we are right in the middle of checking formative understanding for what we discussed last class period on Mendelian inheritance. So I'm going to just show you the questions so you can do this at home. We have a missing student today. So here's question number 1. Remember, pause this if you want to take a minute to discuss it. And then answer and commit your answers, and then we'll go over those answers together. So here's question 1, 2, here's 3. If you can't see this picture, let me make it bigger for you. I don't know if you can still see it, but you've got a father who is big T, and he is a taster, and you have a mother who is little t, and she is a non-taster, and their children are heterozygote. Okay, and here is question 4, maybe, and 5. Remember, pause it so you can commit to your answers. Here's 6. And 7. Here is 8. Here is 9. And here is question 10. I'm going to pause it just for a minute to allow students to finish up. And then we're going to review what we just went over here on monohybrid crosses, and we're going to continue on that, and then eventually we're going to talk about ChiSquare tests and dihybrid crosses. Okay, so we're going to check. Now, as we check, some of you will be tempted just to look through all and just kind of care about yourself, and I understand that need, but I want to address any misconceptions. So stay with us as a team, and we'll go through these together. So concerning the PTC paper, you may or may not have been able to taste it, but tasting is a what? Dominant or a recessive trait? Dominant. Okay? And so here, what is big T, little t? That is a genotype. Okay? Now, genotype is when the genes are presented. And here we have both alleles. We have the big T allele and the little t allele. How could that be a phenotype? What would I have to be able to, what would I do to make a phenotype? Tall, tasting, round, wrinkled, those are phenotypes. It's how the proteins play themselves out. If they give you the alleles, then that is the genotype. If you have the homologous pairs, that is the genotype. Ask your bio buddy, because 2 of you struggled on that. So go ahead, ask them, did you get that one? Do you need assistance?
I did.
You do. I am recording. Do you need me? Here, let me pause. Okay, so, hopefully you checked with your bio buddy. Number 3, what is the genotype of the children? And most of you got that. You said it was a heterozygote because they got a big T from one parent and a little t from another parent. Can you check to see if your bio buddy got that one right? All right. Number 4. What is tasting the PTC paper? That is a phenotype, the ability to taste. Okay? You're describing proteins that are made from the genotype. The ability to taste is a phenotype, right? The genes to taste could either be big T, the genotype, big T, or what could be another one? Big T, little t. Okay? 6 of you struggled, so check if your bio buddy was one of the strugglers. May I assist you? May I help you? Okay? Questions? Keep going.
So if someone has
I can't hear him because you are talking. Go.
If someone has a dominant gene, is there a chance of they cannot taste paper if it's their genes get masked or something?
Yes, if their genes got masked. But don't assume a mutation happens. Go with what happens normally every day. Yeah. Okay, we'll talk about when you question your results. We'll talk about that today. Okay. All right. Number 5 said, is tasting a dominant or recessive trait? It's dominant, right? We talked about it. We reviewed that just a few minutes ago when we talked about people's data. To taste is a dominant trait. Non-tasting is recessive. How would you know that? Well, we've said it multiple times, but in this diagram it says they're a taster and they had big T's. Do you remember that? So you would refer back to that. You would infer from that that it is a dominant trait. Could two parents that are tasters have a child that cannot taste the PTC paper? Tell me, what is the genotype required to be a taster?
Double big T or a
Okay, so big T anything and you're a taster, yes?
Yeah.
So you could have two tasting parents that are this. I don't care what the second letter is, do I? If it's a big T, they're still a taster. If it's a little t, they're still a taster, right? And I know this sounds sexist, but we'll be learning about these. These are pedigrees. I don't know if I've already talked to you about that, but males are always squares and females are always circles. I know, it's terrible. It's been around forever. I can't change that for you. I'm sorry. Zero. I said zero. It is circles. Sorry. All right. I'm not going to explain it to you. All right, come back to me. So if these are tasting and they have a child that cannot taste, so let's say they have a little boy who cannot taste, what is that little boy's genotype? Little t, little t. So is that a possibility?
Yeah.
Yes, if this letter is a little t, if they're heterozygous. So that is possible. Okay, let's look at the next one. Number 7. Concerning your response to number 6, what would the two tasting parents' genotypes be? And we just identified it right here. Let's talk about why E would not work. Could E, let me make E bigger. Just riddle me this. Would that make non-tasting children?
Yes.
Yes. But what was the question? They have to be tasting parents. Okay? Those are not tasting parents. Those are non-tasting parents. Yes, it is true. Non-tasting parents will have non-tasting children. But we specifically said two tasting parents. Right? Check with your bio buddy because there's just a few of you that missed that one and make sure they're okay with it now. All right, and then, okay, we are recording. Number 8, could two non-tasting parents have a child that can taste? All right, let's get our parents back up here. They are non-tasting parents. What is their genotype? Little t, little t. To have a tasting child, they would have to have a big T. Is that ever going to happen? No. Okay, so the answer is no. Check with your bio buddy because 5 of you missed it. Questions to me? Yes?
I thought it was yes because this is probably going to be wrong. But when Mendel did his experiment with peas, the short trait was hidden over a generation.
Exactly. But that short trait was not a dominant trait. It was a recessive trait. So you could hide it. Okay? These are non-tasting parents. Non-tasting, is that dominant or recessive? Recessive. That's like two blue-eyed people having a brown. That would be like two short pea plants having a tall. See that? And what's not going to happen? Does that make sense? All right. Next. It says number 9, concerning your response to number 8, what would two non-tasting parents' genotypes need to be in order to have a tasting child? It cannot work. We just did that. There's nothing that works if they are non-tasting. If we look at, let's just go through some of the answers that people chose. Somebody chose B. It says, are these non-tasting parents? No. First one tastes, and big T, little t tastes. 3 people put C. Do both of these guys taste? Yes. Yes, they do. And it says non-tasting parents. D. First individual, what does that individual do? Taste. And the second one is non-tasting. It said, what would two non-tasting parents be? Somebody picked E. These are non-tasting, but they can't have a tasting child, because to have a tasting child, they have to have a big T. Okay? And let's look, check with your bio buddy, see if they're okay. I'm not making the assumption just because I explained it that you understand it. Right? Check with your bio buddy. Okay? Oh. Number 10. Uh-oh. Half of you missed it. So let's have a discussion about it. If I wanted to find out if a tasting person, okay, so here is my tasting person. Her name is Betty. Hi, Betty. She is a tasting person. She is mad because it tastes bad. Okay? If Betty here is a tasting person, what is her genotype? Big T what?
Something.
Big T something. I don't know what that something is, right? That something could be another big T, or that something could be a small t. I don't know what Betty has. All I know is that she can taste, and she only needs one of the alleles to be that, the tasting allele, right? So who would I need to cross her with? So in order to make a baby, she's got to be with Bill. Bill's always thought Betty was beautiful. Okay? What? She still is tasting. She's feeling not good. What should Bill's genotype be? Little t, because he is a known. If he is a non-taster, we know his genotype, right? We know what his genotype. If somebody has blue eyes, you know their genotype, right? Bill, every single time when they reproduce, every single time, what will Bill give to Betty? A little t. That's a known. Okay? Now, what Betty gives will depend on her genotype. If they have 15 children, because Betty and Bill really like each other, okay? If they have 15 children and all their children are tall, what is Betty's genotype? Big T, big T. Most likely. We'll talk about statistical analysis in just a little bit. Most likely, Betty is big T because all their children are tall. Okay? If, however, they just have one, oh, sorry, I said tall instead of tasting. If just one of their children is a non-taster, then what do we know that Betty is? She only has to have one child. Just one child that doesn't taste, and we know that Betty is a heterozygote. Okay? Check with BioBuddy. Check with BioBuddy.
The single letter is called
All right, moving on. Widow's peak. What is dominant? Widow's peak or straight hairline? Widow's peak is dominant. Let's use the letter W, okay? You need to be ready to do a bunch of crosses quickly, please. So I would like you to cross for me 2 heterozygotes for widow's peak. Give me geno and phenotypes. Try it at home too. I'm going to pause you. Okay, did you set it up like this? 2 heterozygotes. And when you got your data, was it this? Okay, what is the genotype ratio? 1 what? Big W, big W. Go ahead. And? Okay, remember when it's things like W's, P's, and S's, do a little something, something so you know the difference between them, right? Okay, what's the phenotypic ratio? 3 what? Don't say 3 dominants. Tell me 3 widow's peak, 1 non-widow's, which is straight. Okay? I'm going to tell you, when you are doing genetics problems on the AP exam, or for me, if it gives you a genotype or a phenotype, write all that business out. Don't not write it out. Okay? Because you're more likely to make a mistake. You catch it better when you write it out. All right? Do another one for me. Do this one. I'm going to pause you. Hopefully I keep remembering. Okay, did you set it up like this? Did you set it up? I know, I decided I'd use pictures instead. Do you know who that is? Jude Law. He's an actor. Okay. Did you set it up? So it doesn't matter which side you're on, but did you set it up at least like that? Yeah. Okay. So, and did you get something like this? Okay. Give me genotype ratios.
2.
No. What do you always do with ratios? Simplify. Simplify. So now try again. 1. Okay. 2. Excellent. And phenotype ratios.
1 widows, 2
1 widows, 2.
1 straight.
1 straight. I'm not spelling straight. Okay, check with your bio buddy. Easy peasy lemon squeezy.
Easy peasy.
Okay, youngest bio buddy, could you just summarize what you've learned using this diagram or what we've reviewed? Go ahead, youngest bio buddy. You're all over it.
So basically,
I'm not saying those two had sex. I just needed some pictures. Okay. Any questions on that diagram? Pretty straightforward, yeah? Okay, so now we're going to start talking probability. And when you do your POGL, it's all about probability. And when you do your POGL, I highly recommend that you have a calculator and some scratch paper available to you when you do it, okay? When does that POGL due? Friday. At what time?
8:30.
Excellent. All right. So let's talk coins for first. Okay? If I have 1 coin and I flip it, what are the odds that I am going to get heads? Give me an infraction this time. 1/2. And what are the odds I'm going to get tails?
1/2.
Okay. And I have another coin. I'm going to flip it. What are the odds that I'm going to get heads?
1/2.
And tails.
1/2.
Now, I've got a coin in each hand. I'm flipping them at the same time. Catching them. Okay? What are the odds that I get 2 heads? 1/4. How did you get that? Because you multiplied the probability of this coin with that coin. 1/2 times 1/2 is 1/4. Probability I get 2 tails? 1/4. Okay, good job. Now, it so happens that I'm pregnant. I'm not. I'm pregnant, and so is my best friend. She is also pregnant. What are the odds that I give birth to a girl? 1/2. What are the odds that we go into labor at the same time and she also gives birth to a girl?
1/4.
Okay, together, right? But her by herself is 1/2. That we both have girls, it's what? 1/4. That we both have boys, it's
1/4.
How about I have a girl and she has a boy?
1/4.
1/4. Good. I have a girl as a half. She has a boy is a half, right? So we multiply that together, it's 1/4. And vice versa, right? How about this? What are the odds that we have any difference in sex? I have a girl, she has a boy. I have a boy, she has a girl. 1/2. Because now what do? We add all the different ways to do it. If I'm specific, if I say girl, girl, boy, boy, girl, boy, boy, girl, right, that's a specific sequence. But if there's more than one way to get it, if I just say any boy-girl combos, then I add them together, right? Because if I say I have heads, she has tails, 1/2 times 1/2, right, that is 1/4, right? Or I have tails, she has heads. That's another way to get that same thing. We add those together. There's 2 different ways to get it. So we can say there's a 1/2 of a chance we have any kind of boy-girl combo. Yes?
Are there going to be problems where they're like not mutually exclusive? Like,
I'm going to show you. You'll see. I'll show you today. All right. Now, let's revisit the Freckle family. Okay? Let's revisit the Freckle family. Mr. Freckle clearly has freckles. Freckles are dominant over no freckles. So all I know for sure, all I have done is met Mr. Freckle. All I know for sure is he has what? Okay, give me one of his alleles. All I know for sure is he has big F. That's all I know. He meets and marries Mrs. Freckle. She for sure has a... And that's all I know, because she is displaying freckles and I don't know anything about their parents. It would be helpful if I knew something about their parents, but I don't know anything about their parents. They start having children. They have 1 child. They have a 2nd child. They have a 3rd child. This first child has no freckles. What is the genotype of the freckless child? Little f, little f. Assuming Mr. and Mrs. Freckle are loyal to each other, these little f's must have come from them, Mr. and Mr. Freckle. So now it only took one. I don't care about the rest of their children. I do. It only took one freckless child to let me know their genotype. Do you agree with that? Okay, now calculate with your bio buddy. What were the odds that they had 3 freckless children in a row? Calculate with bio buddy. Work it out at home. What are the odds? What are the odds? Okay, now, first thing I would do, it's surprising how often you can make mistakes if you don't jump through a few hoops. The first thing I would do is, oh, Mr. and Mrs. Freckle, let's do their Punnett square. Big F, big F, little f. Freckle child, freckled child, freckled child, freckless child. So what are the odds of having a freckless child? 1/4. And now I have another freckless child. What are the odds of just this freckless child? Because Chance has no memory. What are the odds for this one? 1/4. And what are the odds that this child is also drooling quite a bit and is freckless? Then I ask you the question, what are the odds of 3 freckless children in a row? You're going to say 1 times 1 times 1 is 1. 4 times 4 is? Times 4 is? See how that works? Okay, so we were talking about having 3 freckless children in a row. That would be the odds statistically, since they are both heterozygote for freckles. Perfect. Okay, now let's take a look at these 3 questions that you had for your practice problems. Both a man and a woman are heterozygous for freckles. Oh, we know that story. We just met them. Freckles are dominant over no freckles. What is the chance that their child will have freckles? What's the answer to that? 3/4, right? Go back to your Punnett square. What are the odds that they would have a freckled child? 3 out of 4. Okay, and hopefully you got that. Question number 2. Both you and your sister or brother have attached earlobes. Okay, I don't write anything down yet. I'm just like, okay, fine, good to know. But your parents have unattached earlobes. Unattached earlobes are dominant. Then I start writing things down as soon as I see something like that. So I go, big E anything, that's the dominant one. And what is that equal to? Unattached. Write things down so you don't make mistakes. Okay, it says dominant over attached earlobes. So little e is attached. What are the genotypes of your parents? Okay, both you, your brothers have attached. So attached earlobes is always going to be what? So here is your brother and your sister. They have to be little e because they have what kind of earlobes? Attached. And attached is little e. So what are their parents? Well, here's mom, here's dad. What did it tell me about your parents? But your parents have unattached. To be unattached, you have to at least have what? One big E. But since they had unattached children, or sorry, what kind of you had? Attached. They each have to be a carrier for a little e. Okay, check with BioBuddy. Did they get that one right? Okay, a father has dimples. The mother does not have dimples. Then I keep reading. And all 5 of their children have dimples. Dimples are dominant, okay? So now I know I have a father. A father has dimples. So he has at least one big what? D. I don't know what his second letter is. A father has dimples. The mother does not have dimples. So we're crossing it with what kind of individual? The mother does not have dimples. Homozygous recessive. So she has to be little d. Yes? Okay. Give the, what does it say? All 5 of their children. So they had 5 children. All 5 of their children are what? All 5 of their children have dimples. Okay. All 5 of their children have dimples. Remember, dimples are dominant over no dimples. What did mom give every single time? Little d. So we have a child that's big D, little d, another one, 1, 2, 3, 4. How many children have I had? 5 children. And I know this little d right here came from mom every single time. So all 5 times, dad had to give the big D. Okay? we would predict, that sounded terrible. Just ignore that. If Bettina had not made a face, I would not have even thought about it. Okay, now, we would expect half the time, let's say they only have 4 kids, we would expect half the time Dad would have given the capital letter and half the time given the lowercase letter, right? But it didn't happen every single time here. So we probably think that Dad's genotype is what? Big D, okay? Now, riddle me this. What would be the odds? Okay, if he's big D, then 100% of the time you would expect all the children to be dimpled, yes? I don't give a rip about mom. Doesn't matter, right? Okay, 100% of the time he would have dimpled children. But let's say Dad was a heterozygote. What are the odds that he would have 5 dimpled children if he was a heterozygote? Calculate that. Don't do it out loud. Just, write it or do something there. So, what were the odds of this first child having dimples? Half. Second child? Third, fourth, fifth. 1 times 1 times 1 times 1 times 1 is 1. 2 times 2 is? 4. Times 2 is?
8.
Times 2 is? Times 2 is? So 100% versus 1 over 32. I'm going to go with probably he was big D. Okay? Ask your bio buddy, are you on solid ground? Are you wiggling around a little bit? Can you guys make that the white light I'm learning over there for me? Do you have a thing? Oh, here.
I was laughing today all the time.
It was funny. All right. Ask your question.
So if the,
Listen to the question. It might be yours. Go.
so if the dad was the homozygote dominant and every single one of the children was the heterozygote, how would you write the ratio?
100%. You'd write 100%.
So
100% big D, little d. Or if you're writing. You won't have a ratio when you have the same. You just say what it is.
you want like write a colon line?
No. I mean, you could. You could say 0 this. Right? Okay. All right. Now, next thing. Did I start it? I'm going to have to keep remembering. Yes, I did. Okay. For a certain flower, purple flowers are dominant over white flowers. Purple dominant over white. I have, this is purple. I have a purple flower. I do not know if this purple flower is what or what.
Dominant.
Big P, big P, or big P, little p. I don't know. Why? Because it's displaying the dominant phenotype, right? When you display the dominant phenotype, you do not know what the genotype is, right? You don't know. Okay? So if I have a purple flower, what would be the best cross to make to find out if this purple flower is big P, or big P, little p? What would be the best cross? And that would be a white flower. Oldest bio buddy, it is your turn. Differentiate between the outcomes of these two crosses, and how would that help me determine the genotype of this purple flower?
So if you know because you're white, then it has to something like crazy.
Okay, so remember, this is one of those funky letters. P's, capital, and lowercase look the same, so you got to be careful. Okay, if I don't know, and the reason, how come I know the genotype of the white flower? Tell me that. It's recessive. That's the only way. Blue eyes, I know what you are, right? You're little b, little b. It's not a question. Okay, so if I don't I know the dominant phenotype, but I don't know the genotype, then I would have 2 different outcomes from what I would expect, just like what we did with the dimple family or freckle family, right? If the parent purple plant is big P, all the offspring will be purple. I only need one white flowered plant to know that the parent plant was a heterozygote, yes? That is what's referred to as a test cross. The only ones you need to test are the dominant phenotypes. You do not need to test the recessive phenotypes. Youngest bio buddy, tell your oldest bio buddy, why is that true? Why do you only need to test the dominant phenotypes and you don't need to test the recessive phenotypes? Go. You're only testing what you don't know, right?
Yeah.
So if you're the recessive phenotype, do you know their genotype?
Yes.
Yeah, homozygous recessive. It's the dominant phenotype that you don't know their genotype. That's the one you need to test. Okay, so on your notes, let's look at your notes. I have no idea where you are in your notes, so you might need to help me just a little wee bit. Are we on where? 11.2, law of dominance? That's where I'm at? Okay, law of dominance, one trait or factor can be dominant over another. I didn't give you that right yet? Okay. For example, tall plants are dominant over short plants. Law of segregation. We didn't go over that?
No.
Oh. Okay, let me go back and do law of segregation, but I want to do test cross first, so go down into your notes. Okay, just because that's what we were just talking about. Skip down where it says Mendels Law of probability. You see where I am? Right above 11.3. Got it? Okay, so go down to number 4 where it says test cross. The purpose of a test cross is to determine or confirm the genotype, of a dominant phenotype. To determine or confirm the genotype of a dominant phenotype. Okay, going way back. Law of dominance. Okay? There's 3 laws we're going to know. Law of dominance, one trait or factor can be dominant over another. For example, tall plants are dominant over short plants. Law of segregation. When does segregation take place? You know when? We learned about it. Meiosis, right? So we know that meiosis 1, what gets separated from each other? Homologous pairs. Meiosis 2, what gets separated from each other? What do you call failure to separate? Non-disjunction. Okay? So we know we're going to separate big T from little t. That's the law of segregation. So in summary, each individual has 2 factors, alleles, for each trait. That's right. We went down this deep thing. We tried a bunch of different. I remember that now. Okay. The factors segregate or separate during the formation of gametes. Anaphase 1. Each gamete contains only one factor from each pair of factors. Fertilization gives each new individual 2 factors on homologous pairs for each trait. All right, alleles. Alleles are defined as alternate forms of a gene. Alternate forms of a gene that occur at the same gene locus, a specific location on homologous chromosomes. Alternate forms of a gene that occur at the same gene locus on homologous chromosomes. A dominant allele will mask a recessive allele. There are exceptions. To be homozygous, homo means same, big T, or little t, both alleles the same. Heterozygous different, big T, little t. The genotype is right here. It's these pairs of alleles. Big R, little r, this combination of alleles could maybe make a pea pod round. Okay? Maybe this makes them smile. I don't know. I'm just trying to. This makes them, this combination, they're tall. And this combination makes them green. Okay. Right? That's a combination of those alleles. Now, phenotype is the appearance. It's how the proteins play out. Law of independent assortment. I think we taught, where is that? I know it's somewhere. I think it's hidden. No. never mind. We'll skip over that. That's coming up. That's right. Okay, go to Mendels Law of Probability. Skip to that. We'll come back to independent assortment. Mendels Law of Probability. I would like the slate to take on number 1, and I'd like blue to take on number 2 in the notes. The words you need, slate, is product rule. Product rule, and blue, it's the additive law. Okay, go ahead. Okay, so the product rule of probability is to find out the chance of 2 different events occurring together. You multiply the chance of them occurring independently. Like, what are the odds of heads? And in second coin having heads, it's a half times a half is a fourth. Whereas the sum or additive law rule of probability, it's if 2 different events can occur in any order, like a heads-tail combo or a tails-heads combo. We add up the individual probabilities together. So heads-tail is a fourth, tails-heads is a fourth, so it's a fourth plus a fourth is a half, because there's 2 different ways to get the same results. All right, and then the next part, that chance has no what? Memory. Each offspring has the same probability of inheriting a trait. It is not dependent on the genetics of the offspring before it. It is not dependent on the genetics of the offspring before it. We saw this first freckless child had a fourth of a chance of having no freckles. The second child had just as much of a chance as the first child, right? Okay, still a fourth of a chance. Okay, perfect, perfect, perfect. Okay, I have a question for you. Oh, no. Yeah, here. Okay, now I'm going to show you the questions too so you can see. So pause it so you can work out the questions, all right? Here's question number 1. Here's question number 2. And here is question number 3. I'm going to pause you. Okay, let's give it a go. Yay! Okay. We will want to cross them with a non-taster. Perfect. Okay. Number 2. Yay! Yay! I hope you got those at home because I don't know. Does anybody have any questions? Do you think I need to go? Okay. All right. All right. We're going to take a quick review. This is something you already know. You ready? Remember this. Research hypothesis versus null hypothesis. Remember research if then because. Null is, hey, the results I got, if there's anything that doesn't match it up exactly, it's probably due to what? Chance, right? Due to chance. Okay, so you just have to make sure what is the probability that it would have been due to chance. If it's only going to happen 1% of the time, that's probably not going to be due to chance. There's some other factor that's coming to play there. So for this one, where is that page? Come here, page. Okay, yeah, that's good. Yay. Okay, thank you. Very dramatic. All right. So usually in our genetics problems, we're going to use the null hypothesis. So if I predict that there's going to be 3 to 1, let's say, I would have guessed 3 out of 4 of their children would have turned up freckles, and 1 out of 4 of their children had no freckles, right? 3 to 1. Are you with me on that? If I don't get that exactly, okay, I'm going to say, however I'm off, it's probably due to chance. Then I just have to see how far off am I. So I thought we would do one on having children. So I Googled family with multiple children, and I started to make the presentation. And I was going to say, what are the odds that these 2 parents had these children? Yeah, 0 chance because the children are brown and the parents are white. So I don't think there's any probability that they had those children. But I had to leave it because you can see I still did expected and observed. I got really into it. And then I'm like, All right, so let's try a different family.
Whoa!
Okay? A lot of children here. All right. Okay, now that we have mocked the family, let's do a little math here and see if there's something up with Daddy. Okay? So I'm going to assume this is Dad. And I'm going to assume that this is Mom. Are we okay with that?
Yes.
Okay. Now, how many children did Mom and Dad have? 10 children. Okay. So if there are 10 children, hashtag not sustainable, how many would you expect to be girls? How many would you expect to be boys? 5. I agree with you on that. Okay. Now, let's see what is observed. Okay. So this, I'm assuming, is a girl. What's this? Boy.
Boy. Boy.
I'm looking at that belt and the jeans, and there's no flowers. I'm going to say with... I don't care how you dress your kids. We're just going to, go with this binary world. What's this one? Girl. He can never please his father. He's already in therapy. Okay, what about this one? What's this one? She's trouble. She's been running around. She's currently pregnant. You just can't tell. And this one. Overachiever. Always trying to please Daddy. Okay, here we go. Now, I just had to give him some personalities. All right. He can't read. All right. Yeah. Tell me, what did we... Sorry, it's a long day. What did we observe? How many girls do we have? 3. We have 3 girls. We observe 3 girls. How many boys do we observe? 7. How many? 7. Okay. Now, just to move things along, I'm going to do it with you this time, but I expect you to be able to do it on your own because I'm going to give you several practice problems. So you know that I am going to want to do, look at the numbers because I'm going to forget them. You got them?
Yeah.
Got the numbers. Okay. So I'm going to want to do the chi-square test. You remember your chi-square test. Of course you do. All right. So in this case, okay, how many girls did I observe? 3. How many did I expect? 5. I'm going to square that over what? 5. Okay. How many boys do I have?
7.
How many did I expect?
5.
Remember, I'm giving you easy ones. So when I give you hard ones, you can do them. Okay. Over what?
5.
Okay. 3 minus 5 is 2. 2 squared is 4. Get out your calculators. 4 over 5 plus 7 minus 5 is 2. 2 squared is 4. Man, it looks the same because there's only 2 choices. 4 divided by 5. 4 divided by, what do you have?
1.6.
What is it?
1.6.
Oh, give me this individually just so we have them.
1.8.
Are we good now?
Yeah.
Okay, chi-squared value is 1.6. Now, remember when we do chi-square tests, chi-squaredness, how many degrees of freedom do we have? 1 less than the number of variables that we have, okay, or outcomes or phenotypes that we have. So I had 2 phenotypes, boy, girl. So I take away 1, because if you're not a boy, then you are in a binary world. All right, so I have 1 degree of freedom. What was our number that we were looking at? 1 point what? 1.6. So 1 degree of freedom, so I'm in this row. 1 point, so it's somewhere between 20 and 50% of the time. So is that okay? Is there something up with this family, or can I accept my null hypothesis that they had a normal inheritance of boy and girl? Normal. It's not outside the realm, right? That's acceptable to me. All right, now let's try another one. We're warming up here. Okay, here's another situation. I have a prediction for a genetic cross. My prediction, it doesn't matter if it's genotype or phenotype, I just predicted 3 to 1. That's what I predicted, okay, for whatever the thing is. But my data shows 70 to 30. Okay, is that okay? My prediction, my null here, is that I would get 3 to 1. Okay, 3 to 1. My observed that I got was what? 70 to 30. Is that okay? Do a chi-square test, please, and give me your chi-squared value. Okay, I'm going to ask you to pause whatever your calculations are for a minute because I want to ask you something. Okay, pause and look up here. There's a reason I'm asking you to pause because I don't want you to go down a dark hole. Okay, if I say 3 to 1, first of all, how many total do I have right here? I have how many? 100. I have 100, right? So if I expect 3 to 1, right, that means 3 out of 4 and 1 out of 4. Do you agree with me on that? So what is 3/4 of 100? 75. So that's what I expected, 75 and 25. That was my expected. Did you go down that road or did you go down the wrong road? Okay, self-correct, fix, and go. If you are still working, feel free to work. I'm going to start doing the math up here. If you want to check your math, look up. If you're still working, then just keep working. Okay, are you just at least that far? You at least that far?
Yeah.
Okay, all right. So 70 minus observed minus expected, that's 5. 5 times 5, because I'm going to square it, is 25. 25 over 75. Can somebody give me that number? 0.333, right? Okay. 30 minus 25 is 5. 5 times 5 is 25. 25 over 25 is 1. So my number here is 1.33. Does that sound like a big chi-squared value? No. So let me get you in your right mindset every time you do one of these chi-squares. If your number is little, then you are probably spot on with your predictions. If your number is big, then you are off on your predictions. Okay? Do not be surprised in the College Board if they write a null hypothesis that is wrong. They want you to do the data and discover that the null hypothesis is wrong. Because sometimes they'll be like, here's the data, it's a null hypothesis. They'll ask you a question and you're like, that's not the right null hypothesis, you shouldn't be writing that. They might do that. They may pick a wrong one just to kind of see if you understand it. So just be prepared for that, I'm telling you. So now I go to the table. How many degrees of freedom do I have? 1, because I only had 2 choices here, right? You don't know what it was, but I only had 2 choices. So I'm going to look, 1 degree of freedom. I'm in the first row, 1.3. That would happen between 20 and, where am I at? 20 and 50% of the time. Yes? Okay. So is that acceptable data?
Yes.
Okay. Now, I'm going to do a different experiment. Doing a behavior lab with roly-polies. Okay? And I am telling you, my null hypothesis is roly-polies do not care if they are in the light or in the dark. So if they're given a choice chamber and they have a light side and a dark side in their choice chamber, they don't care which way they go. That is my null hypothesis. This is my data. This is my data right here. Can I support my null hypothesis or not? And please calculate your chi-square value. Can I support my hypothesis or not? And please give me your chi-square value. Okay, and I'm going to pause you. How many roly-polies did I have? I had 50 roly-polies. If I have 50 roly-polies and I'm telling you they don't care whether they're in the light or the dark, then I would expect, as far as my choice chambers go, what would I expect? I'd expect 25 in the dark, and I'd expect 25 in the light. Do you agree with that? Yes. Did I get that as a result? No. So when I do my chi-squared value, I have observed 40 in the dark. I expected 25 over 25 squared plus 10. Okay. 40 minus 25 is 15, right? 15 squared is 225 or something like that. What is it? 225. 225 divided by 25 is 9. This math is the same. 9. What is our number? 18. That sounds like a big number to me. Okay? Right there, I don't even need to look at the chart. Okay? Because now I can tell you I'm probably going to be disproving my null. Let's look. I had light, dark. So how many degrees of freedom? 1 degree of freedom. So let me look at this. 1 degree of, where is that number? Oh, okay, because that would happen less than 1% of the time. So yeah, no. Okay, this means what? This means I'm going to reject my null. And I need to support an alternative hypothesis. My research hypothesis, which is if I vary the light, then the roly-poly behavior will be altered, right? Because they obviously prefer the dark. Okay? Notice I gave you this in the presentation. It's already set up. Reject, fail to reject. Do you see that? Okay, but I wanted you to have good examples of data of when to reject or when not to reject. Okay, now, just because you understand how to do a chi-square test, you still have to figure out what the expected is sometimes. They will just give you data. They will ask you to write your own null hypothesis, come up with your own predictions, and then check it. Here is your example. Read through this. What would your null hypothesis be as far as what you expected? Read through it. I'm going to pause this for a minute. Okay, so come back to me. First thing you need to do is just don't get caught up in the numbers. Just understand the big picture. What do I have mating with each other? Lizards. Tell me about it. Give me a description of these two lizards. So black eye lizards. They're having sex. They have children. Curious, how many children do they have? I made it easy for you. Do not expect that on the AP exam. Okay? They have 100 children. Okay? Now, 2 black-eyed lizards have children. Some of their children, in fact, a lot of their children have what color eyes? Black. Oh, but now we have a new trait coming up. What's the new trait? Yellow eyes. Oh, all of a sudden yellow. What are some examples of where 2 parents have one trait, but a few of their children get some other trait? Where have we seen that before? 2 heterozygotes having sex, right? 2 freckled parents. So that would be my hypothesis, is this is just a regular autosomal dominant trait, and I would expect that black eyes are dominant over what? Yellow eyes. Now, let's take an alternative to that. What if you said, well, I think yellow eyes are dominant? Well, let's think it through. Okay, that would mean black eyes are recessive. You've got 2 recessives, like 2 blue-eyed people. Are they going to have brown-eyed children? No, not unless these lizards are moving around, right? Okay, so I'm going to assume what I've got going on here is I have got, for these eyes, let's just go with B's, I've got a big B, little b crossed with a big B, little b. Right? 2 black lizards that are heterozygous, and they make a lot of black-eyed babies and a few yellow-eyed babies. You okay with that? So then I would make, okay, what were your expected results for black eyes? Well, how many offspring did they have? They had 100 offspring. If they have 100 offspring and you do this Punnett square right here, I am sure at this point you can see this Punnett without even having to do it. I'm just doing it just in cases. Did I start this? Yes, I did. Okay, good. So I would expect This one would have black eyes, black eyes, black eyes, and this one would have yellow eyes. Do you agree? Okay, so I would expect 3/4 of their 100 children to have what color eyes? Black. So that would be what number here? And how many yellow? Okay, do your chi-square test. I'm sure you can from here on out. Okay, so if we were going to chi-square test this, maybe I can do it real tiny right here. What did we observe? 72 minus 75 squared over what? Okay, plus, what's my next one? 28 minus 25 squared over, oh, thank you, you're right. See, that was a dumb mistake. Okay, did you do the math? What'd you get? Ah, what kind of number is that? Little number. So what do we think about our null hypothesis? It's good. Okay, then it's good. Yes?
So if you were to write a research hypothesis, what would it be?
I can't think of one for that. I guess I would say I can't think in that term because it's just wrong. I could tell you a wrong. I know what it would be. So it's hard for me to write a research hypothesis. But if I can tell you an incorrect null hypothesis, I could say an incorrect null hypothesis would be that yellow eyes are dominant over black eyes. That would be an incorrect null hypothesis. Okay? I could do it like that. We'll level it up and then we'll see. Now, I have a question to ask you. I have a question. Here it is. It's very, very difficult. One big question.
I don't know.
If your answer is yes, can you tell your bio buddy, talk about it? What does independent assortment, what is that about? That's what I want you to do. Okay, that's all the time I have for you. Now come back to me. Independent assortment, that takes place during what kind of cell division? Meiosis. Meiosis, because you're getting ready for sex. Meiosis 1, we separate the homologous pairs. Meiosis 2, we separate the sister chromatids. Perfect. Okay, so if we are doing meiosis 1, when do you do synapsis? Prophase 1. Good. When do you do crossing over? When? Prophase 1. Okay. Now, when do you line up in the middle? And that's when we talk about independent assortment, right? Because each homologous pair can line up independently of the other one. Do you agree with me on that? Yeah. Okay. So now I'm just showing you two homologous pairs lining up. Homologous pair dealing with B's and homologous pair dealing with E's. Can you see those letters on there? Here, I'm making them slightly bigger for you if you cannot. Can you see those letters? Yes. What is this individual's genotype? Did you say that? Yeah. Okay. All right. Okay. Big B and a little b, and a big E and a little e. You always write the dominants before the recessive. You okay with that? Okay. If they separate like this, if they line up this way and separate, okay, then all of these gametes will have the what? All of these gametes will have the little b and the big E. And all of these gametes will have a and a little e. But is that the only way they can line up? No. Oh, okay. Okay, so if they lined up this way, what would all these gametes have? And what? Little e, and these would have and big E. Okay, now, you can't do that. He's laughing at my Okay, what would happen, though, we can't do this for every single time, is draw out little chromosomes and big other things, bring the other things around. So let me show you another way to do that. Do you remember FOIL? Yes. Some of you learned FOIL? Yes. Okay, all of you learned FOIL? Last class they told me they didn't know FOIL. FOIL, first, outside, inside, last. Distribution, right? Okay, come back to me. Come back, guys. I don't have a lot of time and I want to teach you. Okay, so if I just did the first two, then I would get a big B and a big E. Do I have that? Yep, check. Okay. If I did outside, I would have a excellent. If I did inside, I would have excellent. And if I did last, I would have, see how I did that? You just have to look at all the probabilities of what you could possibly have. Is this a bigger Punnett square? Yes, it is. All right, time to move on. Now, here are some peas having sex. Okay? This one over here is a true breeding round yellow pea plant. This one over here, tell me what this one is. True breeding what? Wrinkled and, yeah, shriveled or wrinkled and green. Now, tell me, what are all the different gametes that this parent could make? One. There's only one option. What is the one option? Big R and, you see, you can foil that all day long. You're going to get the same four things, right? Okay. How many options here? Okay. So if this is sperm and it fertilizes this egg, okay, this is the parent, the F1's genotype. What will the F1's genotype be? What? Not phenotype, genotype. Big R what? Homologous pairs, people. Homologous pairs. Big R, little r. Big Y. You with me on that? You got to write in homologous pairs because they reunited, right? During fertilization. You okay? Could you very quickly tell me what the possible gametes, if I crossed two F1s with each other, would you be able to tell me that? Yeah. Okay. Are you ready to tell me that? Okay. Okay, so tell me the gametes, please. Big R. We just did this. Big R, big R? Oh, can I have a big R, big R? No, what law says I can't have a big R, big R? Law of segregation. Boom, shakalaka. Law of segregation, because the R's will be separated from each other, yes? Okay, so this one will be what? Big R.
Why?
Law of segregation.
You can't have two
You
R's.
can't have two R's. The R's will be separated. Anaphase 1, we're separating the R's. You ready? Big R. Next. Big R. Next. Next. The struggle is real. Okay? Over here.
Same thing.
Yeah, I kind of want you to say it again because you worry me. Okay. All right. Let's do the next thing. Yes. Let's put this, if this and this get together, what is the genotype? Be specific. Homologous pairs, people. Genotype. Try again. What is this one? Okay, now, do you see the two different genotypes? Do they have the same phenotypes? Because this one's going to be what? Don't say that. Give me the phenotype. Round, yellow. Okay. Round and yellow. What's this one going to be?
Round and yellow.
Can you think of a pattern for this whole row? Why? Yes, because right here, all of them are going, guys, can I just have you focus here? Because all of them are going to get the what? Big R and the big Y. Okay? So they're all going to be round and yellow. I don't even have to look at the rest of those. I already know. Okay? Can you see anywhere else I can do that? Yes, because we have round and yellow right here. Perfect, perfect, perfect. Okay? Now let's check it. Let's look at this box right here. Put them together.
It's round and green.
Round and green. Good. Oh, caca. Sorry. What's this one?
Round and yellow.
Do you agree?
Yes.
And this one?
Round and green.
Round and green. Okay, what about this one?
Round and yellow.
I agree. What's this one?
Round.
Wrinkled and what? Go away, man. Wrinkled and yellow. What's this one?
Wrinkled.
Okay. And what's this one? Round and what?
Green.
Oh, it's that one.
Round, green. What's this one? Okay, wrinkled and go away, man.
I was calling it shriveled.
Wrinkled and yellow. What's this one? Are you good? Now, hang on to that. There are a lot of different genotypes underneath those P's, right? Okay, I'm not concerned about right now. Right now, I just want to know the phenotype ratio. What's the most dominant thing here?
Round and yellow.
And how many of those do I have? 9. Okay, how about round and green?
3.
How about wrinkled and yellow?
3.
And then what's the most recessive?
1.
Good. 9 to 3 to 3 to 1. When you do a dihybrid cross, dihybrid, R's and Y's, 2 hybrids, cross with another dihybrid, your phenotypic expected null hypothesis ratios will be 9 to 3 to 3 to 1, right? But what if I asked you to do a trihybrid cross? More boxes? Yeah, 64 boxes.
I don't even.
No. So I'm going to show you an easy peasy way to do it. Are you ready? Okay. So let me just get a fresh sheet. This was our cross. Our dihybrid cross. Do you agree with that? Okay. First thing you need to do is cross individual punnets. Now some of you can already see this. So just cross just your R's. Just worry about your R's. Do you agree with that? Yeah. Okay? Now, it's going to look a lot the same because they're a dihybrid. Now cross just your Y's. Are you with me on that? Yes. Okay. Now, when we looked at this ratio, we said 9 to 3 to 3 to 1. What did the 9 represent? So the dominant genotype, what would the dominant genotype have to be for them both to be round and yellow? Give it to me. Big R.
Big Y.
It's a genotype, so you better give me 2. So it's big R anything and
Big Y anything.
okay, you agree? Oh, crud. Big Y anything. Do you agree with that?
Yes.
All right, so now let's take it back to here. So if I wanted to know how many are big R anything, big Y anything, laws of probability, how many of them will be big R anything?
3/4.
How many of them will be big Y anything? 3 times 3 is?
9.
4 times 4 is?
16.
9/16. How many squares do we have over here? 16. How many of them are round and yellow? 9. How many were recessive? Little r, little y, little y. 1/4. 1/4. 1 times 1 is 1. 4 times 4 is 16. See how I did that? I don't know. All right, now. Okay, leveling up. I have some corn for you. How many different phenotypes do you see in this corn? 4. You know that because I labeled them A, B, C, D. Okay? What do you think is the most dominant? Which is what? Purple and smooth. Then that would be big P anything, big S anything, right? Purple and smooth. This purple would be big P anything and little s, right? How many would I expect of those? 3. How about here? This would be 9/16, 3/16. How about yellow smooth? 3/16. How about yellow and wrinkled? 1/16. So I could predict, if I could count all of these up, I did. There's 70. Okay? I could predict how many I think are purple and smooth. What would I have to do? 9 divided by 16 times 70. Are you good with that? Okay, I did that. Okay, I went through and labeled every one of those P's. I counted them up. I did round, so you might not have a whole number here. Okay, the purple is the expected. Okay, the different colors are the observed. Could you do a ChiSquare test to see if this Indian corn, Happy Thanksgiving, would fall underneath our null hypothesis of a dihybrid cross of 9 to 3 to 3 to 1? Yes. You need to take a picture of this because before I see you next time, I want you to work it out. This is not for your debrief. I have 2 other pictures for your debrief. Just take a picture of this. You can work it out on a piece of paper. It is not a matter of it's your understanding, so you understand it. Do not make this busy work. It does you no good to copy somebody else's work or look at somebody else's work until you've done your own work to see if you can do this for the AP exam. It's not worth anything other than your integrity. Okay? So do this. Now, for your debrief, I have 2 sets of questions. Here is question set 1. Okay, and here is question set 2. It looks like your other one, but it's not. Okay, so do those 2 pages in your, yes. Go back to the corn because it's so beautiful.
So all 3 of them are the debrief?
No, this one you can just do on a piece of scratch paper, but put the ones that are typed in your debrief. It's not for funsies, these are for practices. Okay? Yeah. Here's a second, let me stop this. Okay, so work on these problems. We'll talk about them next time.